Awesome. Looks like we're all set to go. Let's get started. Hello, everybody. A very warm welcome. Good afternoon. I am Himani. I'm the brand marketing manager here at Cambrian Bioworks, and I'll be your host for today's session. We have an exciting topic for today. We'll be covering whole exome sequencing workflows and how one of India's largest diagnostic labs scaled their sequencing throughput, the challenges that they faced, and the strategies that they adopted to maintain that clinical grade quality even at a large scale. If this is your first time joining one of our monthly webinar sessions, a very warm welcome. You will see a QR code on the screen. It'll take you to our events page where you can subscribe and stay in the loop for our upcoming webinars. We've started this series to create a space for professionals in molecular labs where they can learn from real-world experiences, swap insights, and also connect with others in the community. So far, we have covered topics including stool DNA extraction for microbiome research, selecting the right beads for NGS library preparation, and automating DNA or RNA extractions in clinical settings. We aim to make every session filled with actionable insights to challenges that a lab might face. Let me quickly walk you through what we've got lined up for today. First, we'll begin with the basics, how a typical whole exome sequencing workflow functions, and how lab layouts need to evolve as the sample volume scales. Then we'll talk about common bottlenecks labs face when trying to move to a high throughput load. After that, you'll hear from Strand Life Sciences, their first-hand experiences, what worked, what didn't, and also how they optimized to get the whole exome sequencing running at the scale that they are today. We'll then do a deep dive into one of the key process of this entire workflow, which is extractions. Specifically, how automation systems helped solve several recurring pain points during the scale-up journey. We'll run about 40 minutes of the content, and then we'll have the floor open to a live question and answer session. Let me quickly introduce you to our speakers for the day. First, you'll be hearing from Diksha Suresh. She is the product marketing manager at Cambrian Bioworks. She works closely with our clients in diagnostic labs to understand and optimize their workflow. So she's seen a lot of labs go through that scale-up stage and has some interesting insights to cover. Then we have Ms. Vijayashree. She is the senior scientist at Strand Life Sciences. She has been with Strand for over a decade. Her team has built one of the largest whole exome sequencing operations in the country, and she'll be walking us through that journey, from early challenges to large-scale success, with real metrics and lessons learned along the way. With that, I'll hand it over to Diksha to get us started. Over to you, Diksha.
So I will be taking you through the basics and the fundamentals of a whole exome sequencing workflow and what we have generally seen over many labs that we have visited over the past few years. So to just quickly dive into our topics for today, I'm sure that most of you who have joined in today are already aware of what whole exome sequencing is and must be even practicing it on the daily. However, for the people who just want a quick introduction, whole exome sequencing essentially focuses on the protein coding regions of your genome, which are the exons. This process, it identifies any variations that are in your exome, and this is essentially to diagnose any genetic conditions or study the genetic basis of any other diseases that may occur. In whole exome sequencing, we cover about 20,000 genes in the process. However, there are key differences between exome sequencing and a targeted gene panel. So in whole exome sequencing, while we cover 20,000 genes, in clinical exome sequencing, for example, we will only cover the genes that have certain clinical significance. So this, from 20,000, the number boils down to about 3,000 to 6,000 genes. And if you're going even further for specific disease panels, for example, a lung cancer panel, there might be a much more condensed version of the gene analysis that we'll do. So that number even further boils down to about 10 to 500 genes. With the more you go deeper into it, the more you, lessen the number of genes that you are covering, you can increase the depth of sequencing, and it is also the most cost effective. Now, coming into the whole exome sequencing workflow, let's just take a look at how the typical stages go through in this entire workflow. So firstly, you have your DNA extraction after you receive the sample. DNA is extracted using your bead-based or your column-based methods. However, some studies have proven that bead-based systems are a lot more gentler on your DNA while extracting them. So for workflows like sequencing, it results in retaining all the information that is present. So at this step, there is a QC that is also done, generally with your qubit or your nanodrop, and also a tape station bioanalyzer in some cases to check the DINs or the RIN value of your extracted nucleic acids. So in this case, we ensure that there is enough DNA to meet that threshold to take forward for library prep. Moving ahead, at this point, after your pre-capture procedure is completed, there is a QC again that is performed, which is a quantification using qubit and checking the fragment sizes that are captured at this stage. So typically this lies between 300 to 500 base pairs. And doing a QC at this stage is very important because it detects issues like over or under amplification, or checking for an adapter dimer formation that may have happened. So libraries that fail here, in fact, can be repaired because you're still not too down the line of the workflow. So, or it can even be excluded from pooling going forward. Once the QC is complete and it has all passed, we go into a hybrid capture step, wherein biotinylated probes are added to the mixture, to the reaction mixture, to hybridizing the targeted exonic regions. At this stage, there is some planning that is required if you're running a batch of 24 or 48 or 96 samples, just to make sure that the hybridization is synchronized and there is even pooling that can be done later. Once hybridization is complete, which is an overnight process, the post-capture cleanup step is also extremely important. Here, once the QC is done, wherein you check the library concentration with a qubit or a qPCR, you can also measure the capture size profile at this point to check the success of the enrichment process. And once that is complete, there is a normalization and pooling step that is performed. And this is mainly to make sure that all the samples have equal representation during the sequencing process. So normalization can be manual or automated, but it may be one of the last bottlenecks in this entire workflow. The pool is then denatured and it is diluted and loaded onto your flow cell. Generally, skipping or rushing over normalization step can lead to either over-representation or under-representation of some samples. It ends up wasting your flow cell at the end of it. Now, while we have covered all of these steps are generally cordoned off in certain areas or even certain rooms, which we'll be exploring further. And they're also divided between at least 3 or 4 teams of people who take ownership of a particular step, just to ensure that the whole process is more streamlined. Now let's talk about why a thoughtful lab layout is absolutely critical for a whole exome sequencing workflow, especially if you're in a diagnostic or a high throughput setting. So there are mainly 3 core reasons why we want to physically separate these zones in a lab. One would be the efficiency of the entire workflow, of course, and second and most important, the contamination control, and of course, according to your NABL or your CAP standards, for the regulatory compliance, we separate these zones. Now, usually in how these labs are laid out is that there is a sample receiving and accession room. At this stage, there are samples received not just for whole exome sequencing, but for many other tests as well. It can even include pathology if it's a hospital lab. And the samples are received and correctly loaded into the LIMS system. Then comes your DNA extraction and QC, followed by your pre-capture library prep. Until this stage, DNA extraction and your pre-capture, all of that in fact can be combined in one area, as long as one room, as long as there are different areas. However, splitting them into different rooms or different areas is the best practice to go about it, of course. And then there is the sequencing room, And finally, the post-NGS analysis, which we call the dry lab, the bioinformatics part of this entire workflow. So why the library prep, it's extremely essential to split into pre-PCR and post-PCR is because pre-PCR steps generally have steps like we went through, like fragmentation, end repair, indexing, and all of this generally has to occur in a clean environment where the DNA is quite low. Post-PCR, there are amplified libraries, and this contamination would result in QC failures. So I will touch upon this a little more in the next slide. So to summarize, just having a good layout isn't just about of having good equipment or lab space. It's also a reflection of how your workflow handles volume or biosafety or other regulatory expectations. And ultimately, having that unidirectional flow of samples from the accession room all the way up to the sequencing room is extremely essential and important. Like I was mentioning, it is extremely important to split the rooms, especially in your library prep process, for majorly two reasons. The first one, and the most important one, being contamination control. So your pre-capture zone is a clean zone where your DNA is still in low copy. It's not yet enriched. It is very sensitive and hasn't obviously gone through any amplification. In your post-capture zone, on the other hand, is a DNA-rich zone. So at this point, your PCR has been run, you have amplified specific regions, and now your sample contains millions of copies of your target. Now, even a few aerosolized molecules from your post-capture zone, if that makes its way back into the pre-capture zone, it can contaminate the new batches, it can lead to false positives and mix up the barcoding, and of course leads to wasted sequencing runs at the end of it. Target enrichment specificity also, which is your hybridization step, it is highly specific and sensitive. But this can sometimes even be a double-edged sword, because when it's that sensitive and that specific, if the environment isn't clean, then enriched or, on the other hand, degraded libraries that are floating around from earlier runs can bind non-specifically to the probes that are added during the hybridization step. So this means you can see off-target reads, you can see a drop in target coverage, and overall you lose your sequencing efficiency. So it's not just a contamination risk, but it's also a resource and cost problem, right? At the end of it, you'll be burning reagents and also sequencing space on just noise. Now, let's look at a common scenario which we've seen during our customer visits. So in this particular example that we're taking, this is a small low throughput genomics lab that is running multiple assays like whole exome, clinical exome, and small genome panels in a batch-wise manner. So in a typical batch, there may be 24 samples per batch, and they may run one panel at a time. So as you can see, in a batch of 24, DNA extraction first takes place, then your pre-cap and post-cap for whole exome sequencing, followed by the sequencing itself. This entire process has to complete for a lab like this to start with the next panel. So this sequential batching generally tends to happen when firstly there is a limited sequencing capacity. Maybe the customer has a MySeq at this point, or when there is no automation involved in any of these steps. So neither a DNA extraction machine nor a liquid handler for library prep. And when it's a small team, when there are maybe just a few people that are handling the entire process end to end, that causes delays in, and that is mainly the reason why sequential batching happens. So naturally, they tend to run one panel after another from start to finish before resetting the process. So this sometimes can limit flexibility, and it can also act as a bottleneck when one part of this workflow is there is a chokehold on that part, say for extraction. For example, if it is extraction, the TAT can very easily stretch out during this entire process. It also limits your sample intake capacity, and that is the reason why we see that there is a queue forming because of these constraints. However, even in such labs, we have seen that there is a zone separation that still exists. So even in a single room, despite the small size, there are dedicated zones and dedicated laminar airflow hoods that are made for pre-cap and post-cap workflows. So this prevents contamination again. Now let's look at how things shift a little bit when you move from a low throughput setup to a high throughput setup. In this kind of environment, the layout is modular, and since they see hundreds of samples a week, it is very essential to batch it in a slightly different way. So in this process, there are zones that enable parallel processing. So there may be a dedicated DNA extraction room and a dedicated pre-capture room, a dedicated post-capture room. This streamlines the process a little more, and DNA extraction and such potential bottlenecks are often automated, which makes the workflow much easier to go through. And at this stage, we have also observed that after DNA extraction, say for 96 samples, and pre-capture at this point is done together for all the samples regardless of the panel. Once pre-capture and barcoding is complete, it can be stored at a cold temperature, post which, post-hybridization, the post-capture of those particular panels happen. And finally, the normalization and pooling for all the samples across different panels also take place. So this enables a batching not just by workflow, but also by panel. And it is sort of, I want to say, panel agnostic, in the sense that pre-capture can still happen for all the samples, and it can still be barcoded and stored in a particular condition, and then the next team can pick it up. So multiple teams are also usually involved in this process. So there is process ownership for each team, and batching happens not just by panel, but also by volume. So here, batches are not always panel specific. So pools, like I mentioned, always can come from different panels, but the sample to report time here is more predictable, because you're not waiting to fill up a 96 well plate or fill up 96 samples before a single panel runs. Now, how we sort of envision this workflow is, we even see that the DNA team itself can not just stop at extraction and quantification, but even go up to completing pre-capture and barcoding. And this can be done with the help of automations like the Beluga, which we will be launching very soon. And it is a liquid handler that can automate DNA extraction, quantification, as well as PCR prep. So with automations like these, you can really equip your team and even give more ownership to the DNA team to take it all the way up to barcoding, post which the panel teams own their own post-capture workflows. They can set up the hybridizations based on the panel's requirement, and the normalization and sequencing can be done by another team. So this is where they take over for those particular steps, and they closely work with the bioinformatics team as well. So this structure, we envision, might give more accountability to each team. It might reduce operational costs, and also better coordination involved in the whole process. Now, I want to touch upon some common bottlenecks that we see across different labs, be it high, medium, or low throughput. Some of them being turnaround time delays, which may be, like I've mentioned earlier, caused by having to wait to fill up a full batch of 96 samples if you have a high throughput automation, or panel-based workflows that prevent pooling your samples across assays. And this can have impact on your turnaround time. QC failures that may happen because of poor DNA quality, or manual pipetting errors, or over or under amplification during PCR. It may also delay, again, feeding into your turnaround time bottleneck that is there. Your high consumable burn, wherein you may run partial batches but using a full kit. For example, I'm sure a lot of you must be familiar if you're using a deep well plate format. Though there are 96 wells, you may still only use up, say, 20 at most. So you're still losing that plastic, losing the combs and other consumables that are involved in it. That we have seen is usually a bottleneck. sample swaps or mislabeling can also lead to a delay in report generation at the end of it. This is mostly, a manual error that may happen, or inconsistency in your limb system. And plate or well mix-ups that also generally tend to happen when there are a lot of samples that are involved. So these bottlenecks are not usually due to bad science or anything like that, but it's mostly a process or an operational challenge. So now I would like to invite Dr. Vijayashree to take over and let us know how Strand solved some of these challenges during their scale-up story. So over to you, madam.
Yeah, thanks Disha, and thanks to Himani as well for introducing me. Strand Life Sciences is a genomics based research and diagnostics company where it is a lab with CAP and Enable accredited in next generation sequencing since 2015. Strand Life Sciences cater services in two segments like genetic testing and bioinformatics. Just coming on to the genetic testing, the services where we are providing in different areas in oncology, where genomic testing is done related to cancer. And coming on to the rare diseases, where the genomic testing is done with uncommon illness like mitochondrial disorder, neuromuscular disorder, cardiac disorder, and so on. The next is the reproductive health, where the genomic testing is done related to reproduction, where at pre-conception stage or prenatal or neonatal. Last but not least is the genomic wellness, where any healthy individual can undergo this testing to know the genomic risk so that precautionary measures can be taken. So the next other segment is the bioinformatics services, where we have in-house developed application, Strand NGS, Strand Omics, and Strand Iris. So the sequenced data are fed into Strand NGS, where it will identify the variants in the inherited diseases, and Strand Iris is where it helps in the cancer treatment. So having this introduction about Strand Life Sciences, uh, I would like to take you through overall the testing process, how it takes place in Strand. Basically, there is a pre-test session which will happen wherever relevant, and the samples will be collected across hospitals, and then the sample receipt happens at the lab through logistics, and the lab processing will take care of extraction, library preparation, and sequencing, followed by bioinformatics, where the variant interpretation will happen, followed by the report, which will be dispatched to the physician. So wherever required, again the post-test session will happen in the process. So now I'll be taking you through the early days where the Strand initiated the wet lab operations in 2013, focusing on mainly with whole exome sequencing and oncology tests. So during those days, the samples what we received were handful of samples where we could process it manually, and which has been taken on to the MySic. So that is where we started with the wet lab. So just moving on to the scaling up, how we made it, how we evolved. So as I already told, we initiated our wet lab in 2013 with the manual extraction and the sequencing happening on MySeq. Uh, since the scale up happened, like we looked at with the automation implementation in the system, so that we can cater to more samples into the extraction, and that is where it happened to make the sequencing in the evaluated sequencing platform on NextSeq. But however, with the automation which we had here, catered to only 2 batches per day, for example, because the time taken on the deck was more, and also the consumables which we are using on the deck, like pipettes, tips, and all, was a cost consuming. So that is where like we have to look at with the different automation, where for the medium throughput automation we looked at and we implemented, wherein it can cater to more samples, not more than samples, but at least more batches of samples which we can do in a day. So that is where we came up with the medium throughput automation called Manta from Cambrian Bioworks. And with this, it could cater to more samples to feeding into the sequencing system. That is where we have a NovaSeq, where high throughput sequencing happening in the lab. So as we scaling up the samples in the pipeline, we come across many challenges during scaling up, where the processing of samples, time taken during processing of samples, and limiting the sequencing capability, and the consumables what we are using could be cost incurred, and the human resources where processing of samples. So these are the challenges which we came across during the scaling up of sample processing in the lab. So as I said in the previous slide, where we got into the medium throughput automation called Manta, which is from Cambrian Bioworks. So we have this automation where it can cater to 32 samples. Initially, when we got the instrumentation here, it was a preloaded protocol where it had off-deck lysis followed by binding and washing and elution on the deck. So, but with the throughput, the samples what we are catering here required the lysis on deck, so that the whole process is being automated on the system. That is where Cambrian, they helped us in tailoring the protocol in such a way that the extraction can happen end to end from lysis till elution of the sample. So this, just to speak up with the Manta, it has a loading tray where the pre-filled cartridges are provided, where just loading the sample onto the cartridge onto the Manta gave a efficient way of extraction on the deck. So this allowed us in the pipeline, where the streamlining can be done with more number of samples getting onto the sequencing. So where NovaSeq is the one where high throughput sequencing, it enabled, actually the Manta instrument enabled for us to take more samples onto the sequencing machine. So with all the streamlining, this is the current whole exome sequencing workflow which we have in the lab currently. So we get major samples is whole blood which we receive from different hospitals across India. On receipt of samples, these samples are accessioned and barcoded and then taken forward into wet lab. So DNA extraction is the first step in the wet lab, which happens on automation Manta. So where the samples can be handled in a high throughput manner, and these samples are then followed with the QC. All the quantification and qualification of DNA samples are performed. All the qualified samples are taken forward in the library preparation, where the DNA undergoes fragmentation followed by ligation of adapters, followed by indexing, pooling, hybridization, and the qualified samples are taken forward into sequencing. The sequence samples are then fed into the strand NGS, where the variants are identified, interpreted, and the reports are generated. So just to touch upon with the QC steps in the workflow, what I have shown in the previous slide. So we are doing QC steps at different levels. Soon after DNA extraction, the DNA samples are quantified and qualified. So the samples extracted on Manta have given really a good yield, which is more than 3 microgram, and which is very much sufficient for further downstream assays. And with the whole exome, it requires just 25 nanogram of sample DNA. And then touching upon with the DNA quality, if you look at the when we checked on the protein contamination, A262 by 280 ratio have given really consistent value between 1.8 and 2, which is the expected value for any DNA to be of a good quality. And coming on to the other parameter is a 260 by 230 ratio, which indicates the organic solvent contamination. However, this will not affect any further downstream assays. The next QC step is followed post library preparation at pre-capture level, where we could see on the tape station the peaks are between 350 and between 350 and 450 base pairs, where it's a good quality library which can be taken forward for sequencing. So that is where whatever the samples extracted on Manta followed by library preparation have given a good quality library which can be taken forward for sequencing. Post sequencing, the samples are analyzed and interpreted, and the reports are generated. A template which is being put up here is a template of report where it includes indication for test, results, and interpretation of summary. So, which will tell if the variants are pathogenic or non-pathogenic. Likely pathogenic, and it is according to the ACMG, American College of Medical Genetics. So it's a comprehensive report with interpretation and recommendations, which has been issued to the physician as well as for the patient. So with all this workflow streamlined and scaling up the things made to achieve in the Strand with the sequencing throughput, making more sequencing throughput with 263%, and also it reduced the turnaround time for processing of samples, and also reduced the cost with the operational and consumable expenses. And of course, yes, no contamination, no mix up of samples, whatever processed on the instrumentation. So these are the key wins which we have achieved with these flows. However, other take home messages where we can see that apart from this, where we need to have sample tracking and data handling as we move to the scalability of the samples. Definitely tracking of samples is very much required, and that is where we can have either robust Google Drive spreadsheets or dedicated LIMS based on the scalability in what level the lab is at low or medium or high throughput. So the next part we look at is the QC to optimize with the critical metrics, where you can have more automated system like fluorescence plate reader to make the things better with the high throughput fashion. And also, last but not least, what I would like to tell is like if any lab is forecasting that more samples are getting into the lab, it is always better to have the decision made in the pipeline to invest onto the automation in the early stage to address time and cost challenges. So, with this, I would like to hand over to Deeksha.
Thank you so much, madam, for all your insights. I'm sure it was very uh, I'm sure it was very nice for all our audience to hear Strand's scale up story. And in fact, at this point, I would even like to mention that Strand was one of the early adopters of Manta. So they were able to give us a lot of feedback, and of course, we were able to optimize a workflow for them. Keeping that in mind, I would just like to spend the next couple of minutes going over how Manta can solve these common bottlenecks that are in your genomics labs. Firstly, if the lab that you are in is a low throughput lab that isn't seeing too many samples in a day, however, you are still offering very high value tests like whole exome sequencing or clinical exome sequencing. And in such cases, if you are seeing a variety of samples, not just whole blood, but might even be FFP tissue or buccal swabs or fresh tissue from a biopsy, you can process all of these samples together on the same run on the Manta. And this is possible by lysing your samples off deck so that you will be able to then load the lysate onto your cartridges and place them all on the same run on the Manta. This again helps your TAT improve, and there won't be any bottlenecks in your extraction part of the workflow as such. Now, if you are a lab that is offering a lot of again the same similar type of test panels, but you are seeing a lot of samples of the same type, for example whole blood, on in this case we can do a lysis on deck, which of course speeds up your process even more. So whether you're a lab dealing with multiple samples or a single type of sample, we do have solutions that can streamline your workflow end to end. Just a few reasons why we have heard even from our lovely customers why they like the Manta is firstly that we operate on pre-filled cartridges which minimize your contamination risks. Now, if you can see on the screen, or if you can see my cursor on the screen where I'm pointing to the tray, the tray in fact is separated by a bit of a distance. So your two samples in fact are not sharing a wall. That reduces your contamination risk by a lot. You're flexible, and one more thing that I would like to highlight about the Manta is the fact that you can run any number of sample types that you have in a day. So if you have a single sample, you can run a single cartridge, all the way up to 32 cartridges in a batch. That flexibility sort of allows you to use an automation rather than waiting for days on end to fill up a 96 well format plate. And on deck, the Manta is only takes about 35 minutes to bind, wash, and elute your nucleic acids. And finally, like I mentioned, having that sample compatibility, not just with one sample, but multiple samples that can be processed, really helps labs that see these kind of samples on the daily. Now, I would like to quote one of our customers, Dr. Harsh, who mentioned that Manta sort of serves like a very great extraction source at the beginning of a workflow for a multitude of applications down the line. So with Manta, if your workflows are molecular oncology or infection control, or if you're a genetics lab that is doing a lot of academic research or gut microbiome testing, biobanking, also all of these applications can be supported by the Manta in the beginning of your workflow. And we do have kits ranging from whole blood, fresh tissue, FFP tissue, viral nucleic acid, which is a DNA and RNA kit, cord blood if you are into prenatal testing, your blood RNA, as well as stool DNA for microbiome sequencing. I'm sure some of you who are here may have attended our webinar last month, which was on stool DNA extraction for microbiome sequencing. So and one more point that I would like to highlight in this stage is that some of the cartridges are in fact compatible with a lot of other sample types like body fluids. So if you are dealing with infectious sample types like pus, sputum, or TB sample types. We do have kits for that as well that go on the Manta. So just to close up this session, firstly, I would really like to thank Dr. Vijayashree for cooperating with us throughout these weeks and putting the slides together, and also for being such a supporter of Cambrian Bioworks. It is because of customers like Strand and Vijayashree Madam that we are able to continue doing the work that we're able to do. So just a huge thank you to Vijayashree Madam and Strand Life Sciences on our behalf. And I think Himani will be taking over just to thank everybody and close the session.
Okay, wonderful. Thank you so much again, Ms. Vijayashree, Diksha, for taking us through the entire whole exome sequencing workflow. It was lovely to have everybody here. Until the next time, thank you.